report will be not very long. Let me introduce myself first. My name is Anton Aleksashkin. I am the first year PhD student in the lab of nanomedicine in Lomonosov Moscow State University, headed by Professor Kabanov. Also, I work as a research fellow in Russian part of Pharmaceutics. And in both jobs, I have a deal with different drug delivery system projects. You know, there are a lot of drug delivery systems, such as liposomes, nanogels, and so on. And for each system, you should choose a correct one to get the most efficient result. And I will tell you today about a system for enzymes. For example, for superoxide dismutase, that is antioxidant enzyme. It helps to reduce toxic effect of superoxide radicals, turning them into hydrogen peroxide and oxygen. Then we could use catalase to dismutase hydrogen peroxide into water and oxygen. In numerous diseases, ranging from neurodegenerative till some types of cancer, injection of additional amount of antioxidant enzymes could be very useful for patient. There are some advantages of using enzymes, in particular in case of acute pathologies like injuries or strokes. That is extremely high efficiency. That is much more than for low molecular antioxidants such as vitamin E, and so on. We have available recombinant producing methods for such enzymes, and they are biocompatible. However, there is one big disadvantage. It's rapid elimination from the body. For example, we have no any enzyme just 5 minutes after injection into bloodstream. To deal with this disadvantage, we can use the main paradigm of drug delivery systems to protect the drug from the organism and the organism from the drug. And to do this, we get nanoscale block RNA enzyme complexes, so-called NanoZYMEs. The original technique was proposed by Professor Kabanov in 1980s. We just get negatively charged protein and positively charged block copolymer, that is polycation polyethylene glycol. Then we mix two solutions, and the block RNA complex is forming. Next, we use cross-linking agents to stabilize our particles. With this technique, we can get nice particles that is about 50 nanometers in diameter. They have low PDI, they almost have neutral charge, but there is one big problem. The synthesis is not efficient in terms of how much product we get, and this is does not allow us to transfer this to to industry. And the main my task in the lab was to develop to improve this technique to let it transfer to industry. And I proposed to add second layer to particles. So we changed polycation to protamine, that is quite smaller than polylysine polyethylene glycol. But next we add negatively charged polyglutamic acid polyethylene glycol. This second layer led us to use more strong cross-linking agents such as glutaraldehyde. And this allow us to get particles that have much more specific activity, and we have much more efficiency of synthesis. But this is only one part of excellence. The main part is that these particles retain enzyme very efficiently in comparison with traditional technique. And of course, to evaluate effect on animals, we firstly conducted in vivo experiments and evaluated cytotoxicity. And for experiments with rats, we used doses in green rectangle. Firstly, we conducted pharmacokinetic experiments on healthy rats, and you can see that with new particles we have dramatically increased AUC and time of half-life, much more than for original particles. As for therapeutic efficiency, we used rat spinal cord injury model. Treatment was performed 30 minutes after injury with single dosed injection of our particles or control. The efficiency was evaluated using MRI and also BBB test that evaluates voluntary movement of rats. And so with much better pharmacokinetic profile, we got much more efficiency of therapy that leads almost full recovery of animals in case of new particles in comparison with traditional method or native SOD. So in conclusion, I want to point out that two-layer technique allows us to reach high efficiency of synthesis, and two-layer nanozymes show remarkable therapeutic effect on the rat spinal cord injury model. Thanks for my colleagues, and thank you for attention.
Thanks very much, Anton. Any questions? And